archaeological experts reveal some incredible finds discovered in caves near the Dead Sea in the Middle East.
Among the treasures, more fragments of the Dead Sea Scrolls.
For more than 70 years, the Dead Sea Scrolls have fascinated historians, archaeologists, and biblical scholars alike. Hidden for nearly 2,000 years inside caves overlooking the Dead Sea, these fragile manuscripts transformed our understanding of the ancient world. But one question remained unanswered: who actually wrote them? Then, an unexpected breakthrough came, not from translating the ancient words, but from studying the animal skin beneath the ink. What scientists discovered challenged decades of accepted history and revealed a story no one saw coming: the DNA hidden beneath the ink. Everything changed in 2012 during what seemed like an ordinary lunch conversation at Tel Aviv University. Molecular biologist Oded Rechavi and biblical scholar Noam Mizrahi were discussing one of the biggest challenges surrounding the Dead Sea Scrolls. Although thousands of manuscript fragments had been recovered, scholars still struggled to determine which pieces originally belonged together. Their conclusions relied largely on handwriting styles, parchment color, thickness, and educated guesswork. During the discussion, Rechavi asked a simple but revolutionary question: what are the scrolls made of? The answer was obvious: animal skin. That realization led to an extraordinary idea. If parchment was made from animal hides, then each scroll might still preserve tiny fragments of ancient DNA. Instead of studying only the ink, scientists could investigate the biological history hidden within the parchment itself. Rechavi soon assembled a team of researchers from Tel Aviv University and the Israel Antiquities Authority to test the theory. Since the priceless scrolls could not be damaged by taking samples, the scientists turned to an unexpected source. Over decades of conservation work, microscopic dust had accumulated inside the scrolls' storage boxes. Hidden within that dust were tiny collagen fibers shed from the parchment, and inside those fibers were traces of ancient DNA. Recovering ancient DNA was extremely difficult because parchment making, aging, ink, and contamination had damaged much of the genetic material. After years of careful testing, scientists confirmed most scrolls were written on sheep parchment. However, several important fragments were made from cowhide, suggesting the scrolls originated from multiple locations rather than one community. The discovery that changed biblical history. The story of the Dead Sea Scrolls began in 1947 with a discovery that forever changed biblical archaeology. A young Bedouin shepherd named Muhammad Ed-Dib was searching for a lost goat among the rocky cliffs overlooking the Dead Sea. Hoping to scare the animal from a nearby cave, he threw a stone inside. Instead of hearing a goat, he was startled by the sound of breaking pottery. Curious, he climbed into the cave and found large clay jars that had remained sealed for centuries. Inside were ancient leather scrolls wrapped in linen, remarkably preserved by the region's hot, dry climate. At first, no one realized the importance of the discovery. Over the next 9 years, archaeologists excavated 11 caves near the ancient settlement of Qumran, uncovering nearly 25,000 fragments from around 1,000 manuscripts. Among them were the oldest known copies of biblical books such as Genesis, Isaiah, Psalms, and Deuteronomy, along with legal texts, hymns, commentaries, community rules, and apocalyptic writings. Most of the manuscripts dated between about 250 BCE and 68 CE, placing them during a crucial period in Jewish history just before the rise of Christianity. Naturally, scholars wanted to know who wrote these remarkable documents and why they had been hidden in caves. For decades, the most widely accepted explanation was that they belonged to the Essenes, a Jewish religious sect described by ancient historians such as Josephus and Pliny the Elder. The nearby settlement of Qumran appeared to match historical descriptions of this isolated community, and several scrolls described strict religious rules, communal living, and expectations of an approaching final judgment. One famous text, the War Scroll, even described a future battle between the Sons of Light and the sons of darkness. The theory seemed convincing and became accepted worldwide. Textbooks, museums, documentaries, and university courses presented the scrolls as the lost library of the Essenes, supposedly hidden before the Roman destruction of Judea in 68 CE. For decades, the mystery appeared solved, but hidden within the parchment itself was evidence that no one had thought to examine. Evidence that modern DNA science would eventually uncover, forcing historians to rethink everything they believed about the origins of the Dead Sea Scrolls. The cowhide that shattered the theory. When researchers analyzed the DNA extracted from the Dead Sea Scrolls, most of the results were exactly what they expected. The majority of the manuscripts had been written on sheep parchment, the most common writing material in ancient Judea. Sheep thrived in the dry climate around Qumran and the Dead Sea, making their hides the natural choice for producing parchment. But among the expected results was one surprising discovery that would challenge decades of accepted scholarship. Several important scroll fragments had been written on cowhide. At first glance, this may not seem significant. However, the discovery posed a major problem. The region surrounding Qumran is an arid desert, where sheep and goats can survive with limited water and sparse vegetation. Cattle, on the other hand, require abundant grazing land and a steady supply of water. Conditions that simply did not exist in the Judean desert. This meant the cowhide parchment had almost certainly been produced elsewhere before being brought to Qumran. The implications were profound. If some manuscripts originated in distant regions, then the Dead Sea Scrolls could no longer be viewed as the library of a single isolated religious community. Instead, they appeared to be a collection of texts gathered from multiple Jewish communities across ancient Judea. Scientists then made another breakthrough by comparing the DNA preserved in different parchment fragments. Since every animal has a unique genetic signature, researchers could determine whether separate pieces came from the same animal or from entirely different ones. This provided scholars with biological evidence far more reliable than handwriting comparisons alone. Similar patterns emerged in other manuscripts, including the Songs of the Sabbath Sacrifice, demonstrating that these sacred writings circulated widely across ancient Judea. As more genetic evidence emerged, the traditional theory of a single Essene library became increasingly difficult to support. Instead, the Dead Sea Scrolls appeared to represent a diverse network of Jewish communities sharing scriptures, religious ideas, and traditions long before they were hidden in the caves of Qumran. A new history, written in DNA. The DNA discoveries did not stop with identifying the animal species used to make the Dead Sea Scrolls. Scientists expanded their research by analyzing nuclear DNA, allowing them to study the relationships between the animals themselves. In several cases, they found that different manuscripts had been made from sheep that were closely related, perhaps members of the same flock. This remarkable finding opened an entirely new way of studying the ancient world. If multiple scrolls came from related animals, they were likely produced in the same workshop, around the same time, and in the same region. At the same time, researchers noticed a different pattern. While some scrolls shared close genetic relationships, others came from completely unrelated animals raised in distant locations. This suggested that the Dead Sea Scrolls were not the product of a single isolated community, but rather a collection assembled from multiple sources over many years. Instead of serving as one sect's private library, the caves near Qumran may have been used to safeguard sacred writings gathered from various Jewish communities during the First Jewish Revolt against Rome. Perhaps the most remarkable aspect of this breakthrough is where the evidence was hidden. For decades, scholars focused on the words written in ink. The real breakthrough came from studying the animal skin beneath those words. Tiny fragments of DNA, preserved for over 2,000 years, revealed a history that archaeology and textual analysis alone could never uncover. Today, the Dead Sea Scrolls are more than ancient manuscripts. They are biological records, preserving the story of the people, animals, and communities that shaped one of history's most important collections of sacred writings. For decades, the Dead Sea Scrolls were viewed through the eyes of historians and theologians alone. Today, genetics has added an entirely new perspective, revealing that these remarkable manuscripts likely originated from multiple communities rather than a single isolated sect. Tiny fragments of DNA hidden within ancient animal skins have reshaped our understanding of biblical history, religious diversity, and the world that existed more than 2,000 years ago. As technology continues to advance, who knows what other secrets may still be waiting to emerge from these extraordinary scrolls? If you enjoyed this story, like and subscribe for more fascinating mysteries from history, archaeology, and science.